How are you? My name is Michelle Connor. I'm your VCS for Living Environment. Today we're going to be looking at the biomagnification lab. Before we start, we're going to go ahead and just do an overview of the Teach Hub lessons. So the first set of slides up until the gray background slides, these are all for teacher uses. They go over notes, any modifications you may need to make for your students, standards, synchronous versus asynchronous work, and these will all be taken off before you give this to your students. So once you delete these off, you'll be starting with this purple banner. This is the question for today. Anytime you see the blue box, that's where the students are going to be able to actually type into. This pink banner is the lesson objectives. Yellow banners are instruction and modeling. The green one is independent work. The orange is check for understanding, and our blue is our wrap-up. These are usually Regents-based questions. So if you notice, there's two different locations you can find the lab. So one is in the student worksheet, and then one is the actual first information page, which is here. So if we're looking at the worksheet from, assume it's a student's view, you can go ahead, click these, open up the lab sheet, open up the data sheet, and then open up the actual simulation. So the first one is the actual directions. It gives the background information. These are the directions for the different trials, as well as the discussion questions that the students are going to need. So you're going to need to make a copy of this for each student. The second sheet is the data table that they're going to need. If you wanted to combine them, you totally can. So they're doing the current DDT contamination in parts per million, and then they're also looking at the rolling mean concentration, and I'll show you where those are. You'll notice there's 3 different trials and then several different organisms that we're going to be looking at. So if we go to the directions, there's 3 trials. There's DDT at contamination 0, medium, and high. So what we're going to do is we're going to set the DDT, for the first one set to 0. The second one, medium, we're going to put it approximately in the middle of the slider. And then the third one is going to be set to the highest. We're going to hit go, and we're going to let it run for approximately 1,000 ticks. It doesn't have to be exactly 1,000, but we want to get close to there. And then we're going to go ahead and record our data and look at the graphs. So if we go into the simulation, it looks like there's a lot going on. It's really not. So all we're really looking at is this is the data that we're going to be looking at. The graph, the DDT sliders down here. So we can go ahead and slide it around. And then there's a reset and a go. And then there's a clear. So we can go ahead and clear it. So we're going to go ahead. So the first one's set to 0. So that one we know. I'm going to show you just how to. We're going to go to the medium level one, which is technically trial 2. We're going to go ahead and slide this to about the middle. So you'll notice you're either here or here. It's up to you. You might even be able to go ahead and just slide it to the middle, as close to the middle as possible. Then we're gonna do is we're gonna hit go. Now, the ticks at the bottom is what we're looking at. This is going to take forever if we left it like this. So you're gonna also have a speed control. So what I'm gonna do is I'm gonna bump the speed up as high as possible. Then when I get closer to 1,000, I'm gonna slow it down so I can catch it a little bit better. And then to stop it, we're gonna go ahead and hit go again. So we're at 300, 400. We're looking down here. 750. going to let it keep running again to about 1,000. And that's about 1,000. So we're going to hit go again, and that, you'll notice 1,000 is right here. So we're going to go ahead and hit stop. So the furthest we got was 1,037, so that's fine. So remember, if we're looking at this, we want to be somewhere between 900 and 1,100, so that's good. Now, we're looking for the rolling mean concentration, which is here. And then we're also looking at the regular means, which are here. So we want to go ahead and record our rolling means and our means, and then what we would do is we go ahead and hit reset, and we'd go ahead and hit clear means to start over. And then we'd be again at the highest level. So you have 0, highest, and middle.